Hi everyone, this is Jared Byrne presenting my paper entitled Live Imaging of Cellular Internalization of Single Colloidal Particles by Combined Label-Free and Fluorescence Total Internal Reflection Microscopy on the behalf of my University of Nottingham colleagues Dritten Vlasilau, Snow Stolnik, Franco Falcone and Mike Somick. The interactions of colloidal particles with cells is of great interest to scientists working in the field of drug delivery, given that the mechanism of cellular internalization of colloids greatly impacts their intracellular trafficking and resultant distribution within the cell. Early attempts at visualizing subcellular structures involved in endocytosis of particles, such as the clathrin-coated pits, were performed using electron-based microscopes. Despite providing unparalleled resolution, the electron methods only provide a snapshot in time of the internalization event. Improved temporal resolution has been achieved using light microscopy techniques such as confocal laser scanning microscopy. However, this technique suffers from the drawbacks of poor spatial resolution compared to the electron techniques and the requirement for fluorescent labeling of the cell and the drug delivery system of interest. In some cases, a new labeling methodology may be required for each unique drug delivery system, which can be costly from both a resource and time perspective. The fluorescent label may also cause phototoxicity to the cell. With this in mind, there is a need to develop a system which can study cell-particle interactions without the need for fluorescent imaging, particularly of the drug delivery system. Recently, we introduced a total internal reflection-based microscope, also known as TIRM, which is capable of the label-free imaging of colloidal particle internalization by living cells. The mechanism of image formation in total internal reflection microscopy can be considered to be akin to frustrated total internal reflection. For a uniform object with relative low refractive index, total internal reflection will occur and the image will appear bright and homogeneous since all the light is evenly reflected. When an object moves into the evanescent field, which either scatters or refracts light into propagating light, energy is removed from the incident beam, resulting in a reduced intensity of the internally reflected light. The label-free mode is thus sensitive to the presence of objects, for example a colloid or a cell membrane, which impinge the evanescent field. In general, the image will appear darker as the cell membrane approaches the surface of the substrate. We've also added a fluorescent arm to the microscope through the addition of a total internal reflection fluorescence channel, also known as TIRF. In this present work, we describe the application of this combined TIRM-TIRF microscope to visualize label-free colloidal particles close to the cell membrane and study their internalization in a living cell. In our experimental setup, the colloid and the cell membrane are unlabeled and imaged by the TIRM arm of the microscope, while the endocytic structure of interest, clathrin, is expressed in genetically modified cells conjugated with green fluorescent protein to the clathrin, which is visualized by TIRF. Panel A in this slide shows a TIRF image of a 3T3 fibroblast cell, whereby discrete green puncta correspond to cellular membrane structures formed by the association of EGFP-Clathrin molecules and clathrin-coated vesicles, which are present within approximately 200 nanometers of the cover slip, i.e. within the imaging depth of this microscope. Panel B shows a TIRM image of the same cell at the same spatial location, showing the cellular membrane within the microscope's imaging depth. Cell adhesion points, lamella structures and the nucleus are visible on this image. Panel C is an overlay of the TIRM and TIRF images from panel A and B and illustrates how combined TIRM-TIRF microscopy can be applied to study an event occurring at the non-labeled cell membrane by non-fluorescent TIRM at high topographical resolution, while concurrently viewing fluorescently labeled cellular structures of interest. On panel A of this image, we show a 3T3 cell expressing EGFP-Clathrin, which was taken using the TIRF channel of our microscope. In panel B, label-free TIRM was used to image 500 nanometer diameter colloids. Overlaid images are presented in panel C, and a magnified section of this image is shown in panel D. The co-localisation of EGFP-Clathrin with the 500 nanometer colloid is represented by the presence of yellow puncta in the overlaid image. Image analysis reveals that there were a total of 35 colloidal particles adhered to the polylysine-coated surface. 30% 37% of these particles are co-localized with EGFP-Clathrin. Panel A shows a series of 12 images captured at intervals of 2 minutes per image frame from a typical TIRM time-lapse sequence. This sequence depicts from top left to bottom right the presence of a 500 nanometer colloidal particle in images 1 to 8, followed by its disappearance between images 8 and 9. The TIRF images in panel B show the EGFP-Clathrin signal captured 100 milliseconds prior to and at the exact same spatial location as the TIRM image. If we follow the images, a gradual increase in the EGFP-Clathrin intensity is evident in images 1 to 8, followed by a loss in the fluorescent signal between images 8 and 9. Panel C is an overlay of the TIRM and TIRF images. An increase in yellow intensity between images 5 and 8 indicates co-localization of EGFP-Clathrin with the colloid. The disappearance of all signals after image 8 indicates that both the particle and clathrin have moved outside the microscope's axial imaging range of approximately 200 nanometers and been internalized by the cell. The correlation of the TIRF-Clathrin signal with the colloidal particle imaged by TIRM suggests that the particle was internalized via clathrin-mediated pathway. The time between each image frame shown on these panels is 2 minutes, which indicates that the time taken from the initial association of clathrin with the particle at the adherent cell membrane to the internalization event is approximately 6 minutes. This image sequence shows the results of a similar study for the internalization of a 1 micron diameter colloid. Panel D shows the disappearance of the particle between image frames 4 and 5, and this was captured using the non-fluorescent TIRM channel. Panel A shows a dispersed clathrin signal throughout all frames. The overlay in panel F demonstrates that there is no co-localization of EGFP-Clathrin with the 1 micron colloid during the particle internalization event, thus indicating that the particle is not internalized by a clathrin-mediated pathway. So in summary, we believe the TIRF microscope is a powerful technique to visualize label-free colloids and specific cellular structures at high spatial and temporal resolution. The TIRF technology has also enabled demonstration of the co-localization of EGFP-Clathrin with unlabeled 500 nanometer colloids and their cellular internalization, and this study indicated a clathrin-mediated route of endocytosis for these colloids. We've also shown a lack of such co-localization for a larger 1 micron colloid, suggesting that the cellular internalization in this case does not occur via a clathrin-mediated pathway. Thank you for listening.